And I am Xinyuan Chen. I am a co-PI on the Compass FME project. Today, it is my great pleasure to introduce Dr. Holly Michael to you as our invited speaker. Holly is the Unidol Fraser Russell Chair in the environment and professor in the Department of Earth Sciences and Civil and Environmental Engineering at the University of Delaware. She is also the director of the Delaware Environmental Institute. Holly got her bachelor's in civil engineering from the University of Notre Dame, and she holds a PhD in hydrology from the Massachusetts Institute of Technology. She is also an associate editor of Water Resources Research, recently served as the Geological Society of America James Thompson Jr. International Distinguished Lecturer. Her research interests include saltwater intrusion, water resources management, contaminant hydrology, coastal hydrogeology, groundwater surface water interactions, and in geostatistics. Some of her current projects include investigating the coastal critical zone, measuring groundwater flow into the estuaries, modeling the groundwater salinization due to climate change. Also evaluating the sustainability of arsenic-safe groundwater in Bangladesh, and then application of experimental economics to groundwater resources. So we can see Holly does a lot of interesting research related to coastal, and I would also like to thank her for serving on our advisory board for the Compass FME project. I met Holly back in 2014 when she served as a reviewer on our other SFA project, and I look forward to learn what's new research she is doing nowadays. Thank you, Holly, and please take it away.
Thanks so much for the really nice introduction. Thanks for having me. I'm excited to be here to talk with all of you. Let me share my screen. And can you see it full screen?
Yes, on my end.
Good, okay. So I'm going to talk today a little bit about some work that we've done in my group on salt marshes. And then I'm going to spend most of the time talking about this new project that we have, an NSF Critical Zone Network project that really, I think there are a lot of parallels between our project and the Compass project, which I'm really excited about. So I'll probably go into the weeds a little bit more than I normally would on the things that we're working on and thinking about. The project has only been underway for a year and a half. So from that project, I'll just show some preliminary data, but really hopefully give you a sense of all of our field activities and our hypotheses and questions. And I would really love to hear from you where you see there might be synergies and if there is potential for collaboration. A lot of what I'm going to show you is not my data, especially in the CZN. It's work of many collaborators, and so hopefully you'll be able to see who is responsible, and I'll do my best to answer questions, but hopefully some from our team will also be on this call and can answer questions. So with that, I'll get underway. I don't think I need to give too much of an introduction to this group. I think, I think we all know why wetlands are important, but just to give a short intro, one thing about coastal wetlands is that they're really complex, and that's, in my view, what makes them really interesting to study. So I'm a physical hydrogeologist, so I'm really interested in the hydrologic complexity. We have fresh water coming from land, we have saltwater inputs through tidal channels, but I'm also very interested in how the physics of the hydrology interact with the ecology. We see very distinct vegetation zonations, of course, in salt marshes, and also with the biogeochemistry. We see hotspots where nutrients are processed and stored, and of course, high rates of carbon burial, which is a really important reason to study salt marshes. So if we look at the carbon burial rate of various terrestrial and coastal ecosystems, salt marshes come out right at the top in terms of their carbon burial rate per unit area. But of course, coastal wetlands are highly vulnerable to climate and human pressures, and they are changing. And so that's what a lot of our work is about. And we're really interested in understanding the links and the feedbacks between all of these complex processes, because that's really, in my view, the key to predicting the future evolution of coastal landscapes and the fluxes between land and sea. So I'll start off, like I said, talking about a project that was the PhD dissertation of my former student, Julia Guimond, who's in January going to be an assistant scientist at WHOI. And collaborators on this project were Angelia Seiferth and Kevin Moffett. And the goal here is to look at physical biogeochemical linkages that impact carbon fluxes and marsh migration. This work was done at the St. John's National Estuarine Research Reserve. This is a different site from our CZN sites, but it's close to our Delaware sites. And zooming in, you can see this is the reserve. Can you all see my cursor? Maybe I could make a pointer here.
Yes.
Okay, I'll do the laser pointer here if it's better. So here's the reserve, and here is our primary site where we are collecting data. And then this is the larger salt marsh, the St. John's River here. This is an overly complicated diagram, and I won't go through all of the components, but many of you, I'm sure, are very familiar with these processes. And the idea is that in a salt marsh, we have hydrologic fluctuations, we have these tidal channels, we have periodic inundation of the various zones in the marsh that affects the groundwater levels, which then affects the redox conditions in the marsh, which often vary both in space and with depth. And then, of course, the biogeochemical processing that's occurring. And in this work, we're particularly interested in carbon fluxes. So what we call horizontal fluxes, which is flux between the marsh platform and the tidal channel in the form of dissolved inorganic and organic carbon, as well as vertical fluxes of CO2. And the major control on that in this environment is the carbon oxidation, so conversion of organic matter in the presence of oxygen to CO2. I won't go into detail on the methods here because I'll talk a lot about our methods, which are similar later on, but for this study, we did hydrologic measurements. We also did some biology and geochemistry. We looked at, these are our fiddler crab burrows, which actually ended up being really important in terms of the hydrology in our study, but today I won't have time to talk about it. If you want to know more, I'll tell you afterward. But so we were looking at the crabs, but also, of course, the carbon and the redox conditions. So to give you just a flavor of the field results that we had, this is a cross section again of a marsh where we have the tidal channel over here on the left, and then different ecological zonations that were identifiable. And the ecological zonations corresponded really well with different hydrologic zonations. So near the creek, what I'm showing here is water table elevation versus time. And the dashed line represents the sediment surface elevation. So you can see that near the creek, the marsh platform is overtopped pretty much every tidal cycle, except really during really low high tides. And then in the periodically inundated area, inundation only occurs on spring neap cycles. So during spring tides, and much the groundwater levels are responding, have much less of a response to the diurnal tides. And then in the interior of the marsh, it's very low elevation, and so it's almost always inundated. So the water level is often above land surface, only occasionally coming under the land surface. We also have multi-level redox probes that I'll again show you a little bit more about later when we talk about the CZN project, but essentially these are platinum electrodes. They're mounted on a fiberglass pipe and inserted into the ground. So we're able to measure continuous redox every 15 minutes at different depths. So the blue is the shallowest, then the green, and the yellow is the deepest. And so you can see that the redox responses are very different in these zones as well. So the black lines in each of these plots is the water level. So this is multiple tidal cycles. And the blue again is the shallowest redox. I think the red goes down to about 40 or 50 centimeters. And so even down to that depth, we see tidally pretty strong fluctuations in redox potential. In the spring neap zone, it's much less responsive in terms of single tidal cycles, but quite responsive on spring neap cycles, but mostly only in the upper elevations. And then in the interior, only the very shallowest probe is ever anoxic. Everything is else, every other depth is generally has very low EH. So biogeochemically, these zonations are functioning very differently. And of course, that has implications for the amount of carbon they can store, and therefore for the horizontal and vertical fluxes. So we consider the near shore or the near creek zone to be moderate to high in carbon storage, low carbon storage or high rates of aerobic respiration in the periodically inundated zone, and then high carbon storage in the very anoxic interior. Then the other thing is that, of course, we see marsh migration happening all along our coastline. So this is starting to make headlines. We see ghost forests, all up and down the East Coast and in many other areas. And we're also seeing agriculture effects, definitely on the Delmarva Peninsula, but also here's a Washington Post article about North Carolina, et cetera. And these impacts are happening not due to two reasons. One is flooding. So, the root zones are becoming inundated either from above or from groundwater levels rising, as well as effects of salt. So this is a burnt out area of a field. This is what we see in many of our field sites. So we're wondering, what are the implications of marsh migration on carbon fluxes and stores? So I showed that we can make a linkage from the field data between the physics and the biogeochemistry in terms of carbon storage. And through that link, then we can just use a physics-based hydrologic model to assess how the carbon storage might change over time with sea level rise. So we've been using hydrogeosphere. It's a 3D coupled surface-subsurface model. It allows for variable saturation. This is the mesh, and here it is looking at it in 3D. This is, let's see if I can get this started. Maybe not. Well, there should be an animation, but I don't know if it's because I have a pointer or not. There we go. Okay. So you just see that tidally, we're getting inundation of both near the creek, but also in the interior as the water level, the groundwater level in the marsh actually responds to the increase in pressure. So not everything comes from the channels, but we also see flooding, groundwater flooding. Okay, so from this modeling, we were able to map out the different zonations within the marsh based on Fourier analysis of the simulated heads and the heads that we observe in the different zones. And so then we could look at the percent area of subtidal, tidal, spring neap, saturated, and upland zones. And so on the x-axis here, we have relative sea level rise, and this is due to actual sea level rise as well as assumptions about sedimentation rates. And so the present day situation is here at 0, where we have within our model domain, a small subtidal zone and a relatively large upland zone. And then as relative sea level increases, you can see that the upland zone decreases as well, and the subtidal zone really increases. And if we then make some correspondence between those zonations and carbon burial rates, we can look at how the carbon accumulation relates to relative sea level rise. So the red is the present day carbon accumulation rate from the 0 relative sea level rise simulation. And you can see that with a moderate amount of sea level rise, actually the sequestration potential increases because you're gaining upland marsh more quickly than you're drowning the marsh. But at some point there's a tipping point, and then you get a reduction in carbon accumulation rate. One thing that we thought was really interesting is that we made 2 different assumptions in our model. One is that in the upland, the groundwater level is able to rise in response to sea level rise. And in the other one, it's not. So this is no change in the upland groundwater level of sea level rise, and this is a rise equal to sea level rise. And so you can see that in the case where, say, you have a topography limited condition, your groundwater level is not able to rise along with sea level in the upland, it actually makes a big difference in the conversion to upland marsh. So, so that was something that we thought was interesting in terms of understanding the whole system hydrology is essential for understanding the marsh hydrology. So the insights from that are that establishing linkages between physical, chemical, and ecological ecosystem components allows the use of mechanistic models to forecast future change. Although I would really love, we'll talk about this later, to do some coupled modeling, maybe in collaboration with all of you. And coastal wetland zonation patterns and marsh channel fluxes, which I didn't talk about, are dynamically linked to relative sea level rise and the terrestrial groundwater table, which highlights the importance, like I said, of the regional hydrology and also the geology in coastal wetland dynamics. So this accounts for tides superimposed on sea level rise, but how do more episodic events contribute to marsh migration? We're not looking at storm surges here. And what are the implications of these processes on nutrient fluxes and ecosystems? So that is the basis for this new coastal critical zone network project. Again, this is an NSF project. It's the follow-on to what was the critical zone observatories. In this case, instead of being single sites as they were before mostly, now they're networks of sites. So we have 6 sites in ours. We also have, I think, 6 different institutions represented. Here is our team. We represent multiple disciplines and multiple institutions. We also have a great team of graduate students and technicians and postdocs working with us. This is us in our natural habitat. So you can see some of our sites here, our forested and agricultural sites. So the idea of this project is that concurrent changes in water and chemical cycling are altering the function of the coastal critical zone. So our thematic cluster, which is our cluster of sites, will quantify the coupled processes and feedbacks that govern the hydrological, ecological, geomorphological, and biogeochemical transformations in the critical zone to understand how shifts in the transition zone will translate to changes in cycling, fluxes, and storage of critical elements at the land-sea margin. So this is our conceptual model, a cross-section from the marsh into a forest in this case. We have slow hydrologic processes like sea level rise that lead to intrusion of the saline groundwater into the upland, that lead to a response of the water table and perhaps groundwater driven flooding. Also infiltration of salt water in this intertidal zone. And then superimposed, we have fast hydrologic processes like high tides and storm surges that push that floodwater inland, cause more salinization and infiltration of salt water. And a conceptual model here, this is salinity versus time, or conversely versus distance. If this is the zone, the salinity zone where plants start to be stressed, These fast processes will put the plants into the stress zone, but if they're just periodic, maybe the plants end up being okay. But if you superimpose the slow processes, then at some point there's going to be a tipping point with forest retreat and crop failure. So that's what this project is built on. I imagine this is quite familiar to many of you. Our overarching hypothesis is that forested and agricultural conversion to marsh is driven by flooding and salinity, leading to changes in fluxes and stores of elements. So we have these fast and slow hydrological processes that are affecting the elevation of land surface and fluxes and stores of carbon. And elevation is also affected by the ecological and geomorphological process, and fluxes and stores, of course, are affected by the biogeochemical processes. And then each of these have many sub-hypotheses, which I won't go into, but essentially one hypothesis is about the hydrological drivers, how the fast and slow processes mediate salinization and flooding. Secondly, landscape processes, how, for example, forest root mortality feeds back with elevation in forests. So as the roots die, they collapse, the elevation decreases. That's a positive feedback with flooding. Whereas in agricultural fields, the vegetation change and the human alterations will dominate the feedbacks. And then lastly, the biogeochemistry. Changes in salinity and redox are going to alter the nutrient and carbon compositions and fluxes, and the hydrologic processes will affect the redox sensitive elemental cycling. And that there will be different dominant processes between the forest and the agricultural sites. So we have a network of 6 sites at 3 basic locations. One is at the tip of the Delmarva near the Virginia coast LTER, where there's highest salinity, ocean exposure, and the lowest topographic gradient. Then up along the Delaware Bay coast in Delaware, we have intermediate salinity, coastal exposure, and the highest topographic gradient. And along the Chesapeake coast, we have the lowest salinity of the 3. It's pretty protected from storms and a medium topographic gradient. At each of these locations, we have 2 sites. One is a marsh forest transition and one is a marsh agriculture transition. This is drone footage from Tyler Messerschmidt from BIMS, who's working with Matt Kerwin. And so this is the Virginia Coast LTER site. As he pans out here, you can start to see how the healthy forest is transitioning into a marginal and transitional forest that eventually transitions to ghost forest and the marsh. And so along each of those transition zones, we have at least 4 subsites in both the forest and the agricultural sites, where we go from healthy to marginal to transition, or healthy forest to healthy marsh, and then marginal and transition in the middle. And at each of these sites, we have a lot of instrumentation. For example, we have wells with conductivity, temperature, depth loggers. We have soil moisture sensors, those multi-depth redox probes. We have pore water samplers and rhizons. We have a host of other instrumentation too, which I'll show you in a second. But first, I'll show you what some of these transitions look like. You already saw the Virginia coast. This is Delaware. These are actually flipped from each other, but the marsh is on the top here, transitioning to healthy crops. And here the marsh is down on the bottom, transitioning up to high forest. These are the Maryland sites, again, sort of flipped. So here's the marsh and into the healthy crop. And this is our Maryland forest site, which is Menai Bay. These are marsh sites and into the high forest. So this is a summary of our instrumentation and measurements and analysis. We've grouped them basically into hydrology, ecology, biogeochemistry, geomorphology, and modeling. And so I'm not going to go through all the details here, but I'll go through some of those next. So in terms of the hydrology, the objective is to characterize these slow and fast processes and link them to the salinization and the water table depth. So like I said, we have surface water and groundwater levels, salinity in wells and streams, slug tests. We're getting stream flow at our ISCOs, our automated stream water samplers. This is just I'm going to show you some example data from each of these. very preliminary. This one is sort of hot off the press. This is the Maryland forest water table elevations, color coded again by their location. So 0 in this case is land surface. And so you can see that in the marsh, land surface is very often inundated. And then as you go up to the high forest, the land surface at 0 is very rarely inundated. Here are a few storms that we had. Hurricane Ida. This is a nor'easter and a high tide. And here are 2 nor'easters right in a row. This is over a period of, I think, about a year. And so you can see that, we really get a strong response in the water levels, as you would expect. These are the well conductivities, so corresponding to salinities, and you can see that it doesn't respond as quickly to these events as we would expect. And so, but what we're doing is looking at these data to understand overtopping and infiltration processes and flushing processes in terms of the salinity. We're also using soil moisture sensors. This is what they look like. They're run by a solar panel and a data logger. This is Danielle Pratt. She's a PhD student working with me. I'll just show you quickly a video of her.
So what are you doing right now?
I'm installing soil moisture sensors.
Oh.
A shallow I've augered down to a foot, and then I just put them in the side of the hole, one on top of the other.
Okay, cool. Let's see.
This is the deep one. We'll go the furthest down to the bottom of the foot. Just slide it in. This has a lot of roots though, so it might be. There we go, just like that.
And then you're gonna backfill the hole into the second one too?
Yeah, and then I'll fill the dirt out. I pulled out the shallow one.
Okay.
So Danielle put in 2 moisture sensors so that at different depths, so that we can figure out if the saturation of the unsaturated zone is happening from below, from just water level rise, water table rise in the groundwater, or from above. Karen Gadan is leading our ecology. She has sap flow sensors installed in 12 loblolly pines at each site. The hypothesis is that tree water use is reduced in more saline locations, which would drive a positive feedback in soil saturation and inundation. But wetter soils could also reduce salinity concentrations, so there are some nonlinearities there. Here are some results from some of her sites. This is the top row is high forest, the bottom row is low forest, and each of these columns is a different tree. And this is from December to February. And so what this shows is that sap flow tends to slow during the winter. And generally we have lower sap flow in the low forest than we do in the high. Of course, there is quite a lot of variability among the sites. She's also using band dendrometers to look at tree growth effects of salinity. So these are installed on the same trees as the sap flux sensors. And both of these will be moved every couple of years, probably to different species, to understand species dependent effects. Stephanie Stotts is also working on ecology. She's doing tree cores. The hypothesis is that tree recruitment is slowed or has ended abruptly at salt affected sites. And so she's trying to reconstruct historical climate or storm events that may have caused cessation of tree recruitment. Matt Kerman is leading the geomorphology. He's measuring millimeter scale changes in soil elevation and thickness at the root zone and linking that to the hydrologic and ecological shifts. So, and he's installed these SET, sediment elevation tables, at each of our subsites. And he's also doing RTK GPS, both deep and shallow SETs, looking at marker horizons. I don't have data yet to show from this because we really need long time series to get strong conclusions, but here they are in the field, installing on the left, and then very carefully measuring, taking care not to affect the land surface elevation during measurement. They're also doing drone surveys. I showed you Tyler's survey, Tyler's drone footage before and after storm events, and pairing imagery with remote sensing and field surveys. This is an example of work that Tyler or that Matt's group has done, looking at regional NDVI trends along elevation, which gives us an idea of how the vegetation is changing. And so at very low elevations, we see sea level driven browning, whereas at higher elevations, we see climate driven greening. Then the biogeochemistry. This is, there's a lot of different biogeochemistry in this project. The hypothesis is that slow and fast hydrological processes, as I said before, will change the salinity and redox, alter the N, P, and carbon in the system, and the fluxes. So we have high temporal resolution redox probes, the mini piezometer arrays, ISCO samplers. We're collecting soil cores and doing incubations. So I mentioned this already, we've been looking at these redox sensors, and so they give us really high resolution temporal data, which is cool. This data is really new, so we really haven't had a chance to analyze it yet, but this is from our Delaware forest site. This is the high forest, mid, low, and marsh, and you can see, so the black is soil water content. So of course, soil water content is quite different. You can really sort of see a transition from the high down to the marsh. And then the depth distribution and temporal distribution, or the dependence of the redox on the soil moisture, is also different at the various sites, and it also varies with depth. But you can see that in many cases, the redox is very dynamic, and so we expect there to be some exciting biogeochemistry happening there. Yo Chin's group is using voltammetry, which is capable of detecting multiple redox active species like oxygen, manganese, iron, including their complexes, different sulfur species in just one scan. And so this is an example of deployment for a different project, but this is in surface water. We're hoping to be able to deploy this in the groundwater. Angelia Seifrith is looking at the chemical signature of the pore water. She has rhizons installed. This is an example of some of her data, along with Sean Fetro, who's a graduate student. This is the salinity along the marsh forest transition in the Virginia coast site. So you can see, as expected, this is the mean salinity is highest at the marsh and lowest in the healthy forest. And he looked at the distribution with depth as well, and you can see that there's a little bit of change with depth in the various zones, but certainly higher salinity in the marsh than in the upland. This is the redox. So overall, the redox conditions tend to increase with distance from the marsh. And we also see some interesting sharp transitions in redox. We think that these are probably as a result, or probably driven mostly by lithology. And pH, the marsh is more basic, while the high forest is more acidic. And again, some interesting depth distributions. Kate Tully and Yo Chin are collecting these ISCO samples and analyzing them for various things. This is an example from Liz Whitney and Yo Chin's group. This shows a correspondence between DOC, or anti-correspondence between DOC and salinity over a tidal cycle. So as the tide goes out, or as the tide rises, the salinity increases. This is the orange. The DOC concentrations tend to decrease. They're also looking at the quality of the carbon. So this is SUVA absorbance. the higher values are more terrestrial, the lower values are more marine. These are the data from the 3 sites, again, over this tidal cycle. And you can see that there's a clear dependence of the quality with the tidal cycle, and where we have more marine during high tide, more marine organic carbon. Angelia Seifert's group is using spectroscopic techniques to measure carbon and other elements in soil cores. And then lastly, quickly, Matt and Sergio and I are working, well, will be, this is sort of in the out years, we'll be working on modeling. And so we're hoping to integrate all of the data that we get and then get process-based understanding and prediction of the hydrological, ecological, and morphological dynamics. And so this might be 3D hydrogeosphere models. This is one of our models that we're doing storm surge inundation and groundwater salinization, but we'd also like to couple in the marsh geomorphology and get more mechanistic feedbacks among the vegetation, the hydrology, and the geomorphology. And so perhaps there are some tools in the models that you all use that we could take advantage of, and I'd love to talk about potential collaborations there. So I will wrap it up and just say that I think, like you, we're interested in all of these aspects of marsh migration and coastal change. We need to consider them together in order to do a good job of predicting changes in chemical cycling and fluxes. And thinking even bigger scale, you know, in the whole coastal zone, I think we really do need to think about the larger scale links and feedbacks in order to predict the future evolution of our landscapes and fluxes. One last thing I want to mention is that we have some budget for research travel awards. So if you know of anybody who may want to work with us, come visit our sites, take samples, we do have some funds to cover those activities. And thank you. I'll stop there.
Thank you very much, Holly, for this very inspiring presentation.